my dearest little champions, I welcome you all to PU2 Brahmastra Series. Let me tell you, today is day 56 and today is the last day of our Brahmastra Series, and we are so happy for the love we have received for this particular series. I hope it really helped you well. From tomorrow, we are going to start off something else which is going to be really useful for you. Let me not break the suspense if it is so. Now, continuing with what I was teaching, today we are going to complete the most important diagrams. This is the part 2 video. Part 1 was published on Wednesday. So if you have not watched it, watch it. It's really important. Okay. And how to remember the diagrams, but before answering that, I want to remind you something else. That is, we have one particular, channel for foundation children as well, especially those who are in 10th grade. So anyone who have their siblings studying in 10th grade and who are worried about how they would be dealing with the board examinations up there, you can ask them to subscribe to this particular channel. We are the teachers up there, so you don't need to worry. If you trust us, you can trust that channel as well. Yeah? Now, whenever you find it difficult to remember the diagram, first thing that you have to do is understand it completely. Okay, understand the concepts behind the diagram, so then it will be easy for you to remember it. For example, in the case, as I always say, in the male reproductive system, if you are aware, the testis is a place where sperms are being made. So you know the first part is the testis, and then comes the accessory ducts, then comes the gland, then comes the external genital organ. So if you are aware about its function, you will be aware about its localities and stuff like that as well. So understand the diagram really well. Once you're very clear with the concept, what you have to do is you have to look at the diagram and then you have to draw it. That is first trial. Okay, see and draw. That is the first thing that you have to do. The second time you are going to draw it without the use of the reference. Do not take the help of your NCERT. Close your NCERT and draw it. Whatever you remember, you draw it. Now, there are chances that you might be forgetting some things. Right. So once you are done with the drawing, what do? You open the NCERT textbook and start labeling it. Once you label, you understand that, oh, this part is not there, which is why I am not able to label it. And you might be able to recollect it much more better, my dear children. And labelings also you are studying in this manner. Now, suppose you are done. Okay. Third time when you are drawing, it's the final drawing that you are having. You are going to keep it unlabeled. Do not label it. Label it after 2 days. In that way, you would be actively recalling, you'd be thinking of the structures again, and it will help you remember things for a longer time, especially when it comes to diagrams. So please make a note of these things. You have to follow this. Yeah. Now let's start. Okay. First, we have human reproduction chapter, my dear children. So over here, the diagrammatic view of the male reproductive system is a very important diagram. You have to remember it. You have to draw it. And as I said, whenever I show you a diagram where you have to draw it in the examination, what do? You put a star mark. So there are diagrams from which questions come. That is something different. These are the diagrams from which you actually have to draw. That you have to practice. That's the only way that is the only way you can remember it better. Okay. So these two structures are really important. Page number 19, page number 29. Mark it right up there. Now this structure I am going to make and I am going to show it to you so that you remember it for a longer time. Okay. So are we ready? Let's draw. Chalo. Okay. Is it there? So first things first, you have testis. So let's make the testis. One is up over here and one is up over here. Okay. This is the epididymis. So inside the testis you have testicular lobules, my dear children. Okay. Each lobule has so many seminiferous, 2 to 3 seminiferous tubule. The seminiferous tubules open to form the tubular erecti, which is not given in any NCERT, but then it forms something like this, which is known as rated testis, which is given in your NCERT. This is what? Rated testis. Now from rated testis, again the sperms are going to travel outside the testis with the help of epididymis. So you have what? You have rated testis, and then this structure, vasa efferentia. Vasa efferentia. This structure that you see over here, it is epididymis. Okay, this is epididymis. Now, from epididymis, there will be another structure which will be coming forward. That is what? Vas deferens. So, from here, it is going to go up. Similarly, from here also, it is going to go up. Okay, that side I'm not going to draw, I'm just going to draw this side. I'm going to show you. This is what? Vas difference. So these are all the accessory ducts, 1, 2, 3 and 4. Fine. Now, there is a urinary bladder in the case of males. So you have to make the urinary bladder. Up over here you have the urinary bladder, which opens to something like this. Cool. Urinary bladder is connected to the kidneys via the ureter. So you have to make the ureter as well. Okay, little difference, little bit of change up over here. Just give me a second. So this vas difference that you're going to make should be coming and coiling around and coming forward like this. Similarly, should be going like that, coming forward like this. Cool. So it's like a shawl. Now, the vas difference which is over here, it is going to get secretion from the first accessory gland, that is the seminal vesicle. Cool. So it is receiving from there also. Now both of them, they are going to form something else together, which is known as ejaculatory duct, and this ejaculatory duct is going to open up here. Similarly, you get it from here also. Okay. Now comes, this is the first accessory gland, the seminal vesicle. Now comes the second accessory gland, which is like donut shaped. It is what? Donut shaped, my dear children. It has a hole in the middle through which the other ejaculatory duct and everything passes through, and also the urethra passes through. Okay. Now the urethra is over here. Let's extend the urethra. Let's suppose it goes up till here. The end of the urethra is known as urethral meatus. What do you call it? You call it urethral meatus, my dear children. Over here, you should be forming the penis. So let's form the penis over here. So this is the penis, and there is an extra, what's a skin sort of structure over here, which is going to be the, where shall I draw it? Just a second. Okay. Sure, I have lost it. Now that picture is not going to come again. Anyways, let's draw from here, since we do not have to waste too much of your time. So we were here, the urethra is here. Okay, the urethra is up over here, and you have the, the penis up over here, and also the glans penis. Okay, and also the glans penis and the urethra have to extend until this particular region. Well, this area that you see, it has an extra layer of skin. Okay, it has an extra layer of skin which is known as foreskin, my dear children. It is known as foreskin. If you are not able to see, I will draw it in purple and show. This is the foreskin that is exclusively present only in the lower region. And now just here there will be two glands, that is the third pair of gland, which is known as bulbourethral gland, and we are done with that. So this is the diagrammatic view of the male reproductive system. Cool. Now over here you have the female reproductive system, which we already do, okay, which we have already seen. It's on page number 29. I hope you know this image. It's very easy. There is nothing difficult up over here. If you still want me to draw, I can definitely draw it. Okay, chalo, let's draw that as well. I hope this time it does not go. So what do? You have a dome shaped structure like this, make hand shaped structure like this here also, and then okay. Now here what you need to do is, basically you can give a covering like this, and here you can draw. This is what? The vagina. This will be what? The cervix, my dear children, or basically the cervical canal. Up over here, you will be having the fimbriae. Here also, if you come around, you will be having the fimbriae. Let's make the fimbriae here as well. Yeah, cool. That side has went too much far, right? So let me just remove that. This time let me just be careful. Okay, works. Now it's fine. Yes. Now over here you have the ligament, and then you have the ovary. Here also you have the ligament and then the ovary that is going to collect things. Over here, this region is known as the uterine fundus region. Okay, and you will be having certain layers. Up over here, the uterine wall is going to be made up over here, and that's how you draw it. This is the fundus. This is what? Okay, just a second. This is what? Fimbriae. Fimbriae is a part of what? Infundibulum. Then you have a broader region known as ampulla. Then you have a narrow region known as isthmus. This is what? The uterus, and with that we complete the female reproductive tract. Easy. It's really easy. You just have to practice it. Okay? So that is the structure. Fine. Chalo. So cervical canal they have mentioned in the gap, and this region, the fleshy shaped structure that you see over here, this becomes the cervix. Okay, just make that particular change in the diagram. Now over here you have this particular diagram to draw as well, which is the structure of the mammary gland. I have drawn it in the Brahmastra series. Just go and watch it once. Okay, it is really important. They might ask you to draw it. Diagrammatic sectional view of mammary gland on page number 30. Put a star mark up over there. Now, schematic representation of spermatogenesis and oogenesis, again something that they have asked to draw, and questions have also come from this. So this is not something that you have to miss. Okay, you have spermatogonia at puberty. Now this spermatogonia undergoes mitosis. It increases its number. You see it increases its number, and now one among them, basically they undergoes differentiation to form the primary spermatocyte. This primary spermatocyte will undergo first meiotic division, my dear children. This is the primary spermatocyte, the blue one. Okay, so you divided by mitosis, one among them is really specialized. It has underwent differentiation also. This is what is the primary spermatocyte. Now this primary spermatocyte undergoes first meiotic division. You have two secondary spermatocyte up over here. This will undergo second meiotic division. You have 4 spermatids. These spermatids will mature or differentiate into spermatozoa, and this process is known as spermiogenesis. This whole process is known as spermatogenesis. Okay, how it is different over here? Here, similarly, oogonia is there, the mother cell. Okay, it undergoes mitosis, it undergoes differentiation. One among them becomes the primary oocyte. Okay, there is too many spermatogonia, similarly too many oogonia is also there. So you have the primary oocyte up over here. Oocyte starts having meiotic division, my dear children. Okay, from birth till puberty, oocyte, primary oocyte is in that particular stage itself. Okay, when it hits puberty, at this point of time, it will complete its division. Okay, the primary oocytes which are there, it is going to complete its division, not all of them, few of them. Okay, and form secondary oocyte and first polar body. So cell division is happening, but one cell is of no use, that is the first polar body. The other cell which is there, it will again divide. That is the second meiosis will happen over there, and you get one ovum and one second polar body. This is again of no use. This is the only cell. So from one primary spermatocyte, what are you getting, my dear children? Only one ovum. One primary oocyte, what are you getting? Only one ovum. Whereas over here, from one primary spermatocyte, you are getting 4 sperms. So that is the difference. So ratio is asked. So 4:1. Basically 4 sperms are being formed from one primary spermatocyte. Over there, one ovum is formed from one primary oocyte. Cool? Works? Chalo. So that is there. Okay? Please remember that. Now here also again, this picture this diagrammatic representation of menstrual cycle. You are not asked to draw and all. Until now there is no question like that, but they will ask you, this is what are these hormones, who release these hormones. So FSH and LH, which you see over here, are released by anterior pituitary gland of the female. Why it's not coming? Yeah, anterior pituitary gland of the female. Then they will ask you, where are these released? In ovaries. They are released in the ovaries. Okay, so these are developing follicles. What happens in this stage, that stage, and everything? How many stages are there? Menstrual phase, then what's the follicular phase, luteal phase, everything. When does ovulation happen? During LH surge. So you should be knowing about all these things. Okay, that is what is the use of this particular picture. So just analyze this picture really well every time question is being asked on menstrual cycle, so do not miss it. It's on page number 34. Cool. Now up over here, embryo formation. If you see embryo formation, they have given different diagrams. They have named also. So first is there is the egg that comes and fuses, and now you get this. The zygote is dividing into forming an embryo. This is morula, my dear children. Where is morula? Is E. E is up over here. So it's towards the end of the oviduct. From here again division will be taking place. It will form blastocyst stage. In this stage implantation takes place. They will ask you in which stage does implantation take place. It is in blastocyst stage, as you can see over here. In blastocyst stage, all the cells that you see have arranged into two layers. The outer layer is trophoblast, the inner layer is inner cell mass. Cool. So that's how the question is being asked. Why it's not going back? Yeah. So mark it. What do? You put a tick mark where you don't have to draw, but you have to understand the diagram really well for the questions. Okay? Chalo. Now, evolution chapter, there is only one very important image that comes, and that is about the experiment being done by Miller, S.L. Miller. Okay, S.L. Miller experiment about chemical evolution. So over here he maintained a very closed atmosphere. Okay, just like how we had in the primitive atmosphere, there was no oxygen. So here also there is no oxygen, my dear children. There is a vacuum pump. As you can see, there is no oxygen. There is boiling water for water vapor. So in the primitive earth also there was water vapor. There was so many other gases like methane, ammonia, okay, and H2, helium, everything was there. So over here you have four. You have to remember the four of them. What is that? Methane, ammonia, and then H2O, and also H2. And it was too much hot in there. Here also they have put the electrodes for making it too hot. So it is 800 degrees Celsius up here in his experiment. And then he has used condenser, things like that. And later everything that it condensed over here, he found that basically with the help of these gases, some organic compounds were being formed, such as amino acids and stuff like that. So it was accumulated up over here. This image is really important. So you have to draw and practice it. Where do you find it? Page number 112. Page number 112. Cool. I hope you do not forget. Chalo. Now reproductive health. In reproductive health, they won't ask you to draw this. They would ask you to identify the image. So over here, if you see, the vas deferens is cut and get tied up. This is vasectomy. Okay. Here the fallopian tube is getting cut and tied up. This is nuss tubectomy. So it is on page number 45, my dear children. Okay, both these images you would be asked to identify. You would be asked to identify this also. What is this? Implant. This is implant. Okay, they release chemicals inside our body, progesterone and estrogen combination chemicals, which would avoid what? Pregnancy. So these are implants. Please make sure you are able to identify it. It's on page number 45 again. So these are just identification images. Should be knowing about that. For example, he can ask you, okay, what is this? How does this type of method help in contraception? So you should be knowing a bit about tubectomy. You should be knowing a bit about vasectomy. You should be knowing a bit about implants. Okay. So that is how the questions are being framed. First of all, you have to identify it. So for identification, you should be very thorough with the diagrams. Cool. Next, so microbes and human welfare. In that there is a biogas plant which is given. The question have come to draw it. So you should be knowing to draw it. So what do? You take it over here. There is a small tank. In this tank you are going to put the waste and everything. For example, cow dung or whatever waste is there. And then this is going to slowly move into the digester tank. This is over here there is no oxygen. Okay, there is no oxygen. So there will be anaerobic bacteria, or basically methanogens. Okay, methanogens are present in the cow dung. So it is going to basically form lots of methane gas. Okay, lots and lots of methane, carbon dioxide and all things. These gases are taken with the help of a pipe to different kitchens and it is used as biogas and stuff like that. And the remaining content which is there, it can be taken up. Okay, usually it is present at the lower level, not at this higher level, but in your NCERT it is given like this. So basically this is being collected and this can be used as fertilizers or manures for trees and plants and stuff like that. So this is a biogas plant. If you understand the concept, you will be able to remember it as well. So I have drawn it when I was teaching microbes in human welfare, so you also have to practice it. Okay, the diagram is here. Make a line, show it like this, make another box. So slanting lines, parallel lines, you can remember like that. Okay, and then a gas line. Cool. Chalo. Biotechnology principle and processes, sticky ends. They will ask you how does sticky end formation occur. So you should be knowing how to form sticky ends. Okay, so please follow this. GAATTC is there, that is eco R1 site. Okay, so over here it cuts between G and A. It is going to cut between G and A up over here and G and A up over here. Similarly, G and A up over there, G and A up over here. So you got a sticky end. When you cut, everything else will be removed. Here also when you make a cut, everything else will be removed. So what do you get, children? You get something like this. Now both this thing that you are going to stick together, it is going to be one unit of DNA. Okay, this is the recombinant DNA. You will be finding this on page number 166. You might be asked to draw, you might not be asked to draw. But this thing again is important, PBR 332, it's a plasmid vector. This can be asked for drawing. This is asked previously, so this can be asked for drawing. Okay, it's not very difficult. Okay, it's not at all difficult actually. Make a circle up over here. Okay, and each plasmid have an ORI site, that is origin of replication, and then it will be having ROP site, that is replication protein site. There is a protein that is very much required for replication process to happen, that is ROP. Okay, now you have tetracycline resistance site over here. Okay, and you have ampicillin resistance site over here. This is an antibiotic resistance site. On the antibiotic resistance site, you have two recognition enzyme sites. What is it? It is BAMH1 and SAL1. This is an enzyme which cuts over here. SAL1 is an enzyme which will be cutting over here. Okay, in this site. Similarly, PVU1 is there and PST1 is there. There is PVU2 which is there in the ROP site. Then there is EcoR1, CLAV1 and HIND3. So how many enzymes are there? 1, 2, 3, 4, 5, 6, 7, 8. 8 recognition sites are there, my dear children, and you have to remember ORI, ROP, ampicillin, tetracycline. Here you have 2, here you have 2, here you have 1, and in the free area also you have 3. That's all. Cool? Just give me a quick second. Okay. Now we have few more pictures which are important. Now this is PCR. Okay, I hope you know that. It's very easy thing. Okay, you. Come on. Yes, it's on page number 172 where you have the DNA strands together. Okay, then it undergoes denaturation where they get separated. Now you do annealing when one primer comes and attach over here and one primer comes and attaches over here. Then you have extension where this primer is going to get extended with the help of polymerase, that is Taq polymerase. And then again you have two strands. So you're amplifying the DNA with the help of polymerase chain reaction, and this is given in 172 page number. On 172 page number, you have bioreactor given on page number 174, and this is important. It has been asked in the form of diagram, so you should be knowing how to make it. So there is a cylinder sort of thing up over here. Okay, you see a cylinder up over here. Then you have to make certain blades. Okay, then you have to make pH acid control, pH control, steam for sterilization and things like that, a motor probably to run this guy, and also a timer if required. It will stop after some time. Okay, then there will be culture broth, and this is tightly packed so that there is no air inside, okay, and everything air, sorry, not air, basically you're providing air and everything, so it should be mixed with the help of this particular thing. It's tightly packed to avoid contamination. That is what I was trying to say. Okay, and there will be a foam breaker, everything, foam and everything can come back and stuff like that, and there will be sterile air that will be put inside so that it can be transported everywhere. You have a impeller as well. Okay, that's all. It's easy. So that is the structure of bioreactor. Suppose situation where you have not studied this, okay, because you didn't feel it's important and you got a question from that, then what big loss it will be. That is why I am saying listen to me very carefully while I am teaching. Okay, so draw it properly. Now what do we have, children? Biotechnology and application, there is only one structure, okay, one picture, structure of insulin, my dear children, given on page number 182. So this is a pro-insulin which has A, B, C, all the peptides. Now in normal insulin you just have A peptide and B peptide. You are going to remove the C peptide. This is pro-insulin, this is not active, this is the actual version of insulin which we have. A and B are only there, the C is being removed. You just have to understand that. Cool, and if you want to remember, you can remember it also. A is there, then B is there, in between what do you have? You have C. So you cut it down, you get A and B separate and C's will be the separate thing. Cool. This is biotechnology and its application, page number 182. Now organism and population, again only one picture, that is given on page number 194, the population growth, logarithmic and exponential graph which is given over there. Know the formula dN by dt is equals to Rn. Okay, R is intrinsic rate of growth factor, and over here you have dN by dt is equals to Rn k minus n by k. What is k, dear children? Carrying capacity. So k is carrying capacity, it has a sigmoid curve, it has a J-shaped curve. So J-shaped curve is for this, that is logarithmic growth. Over here exponential growth, and the other one is for the logarithmic one or the logistic one. Okay, cool. Better you say it as logistic only because it is given in your NCERT. Cool. So this is that, and this will be exponential. Next, ecosystem. In ecosystem you have different things, pyramids given. So pyramid of numbers in grassland, as you can see, the most of the thing is acquired over here, then it goes up. Pyramid of biomass, if you see, it will be again upright only. Pyramid of biomass in the case of terrestrial land is also upright itself. Okay, because why? The plants, they occupy a lot of area, they have lots of biomass, and the organisms which are on it will be smaller in size, which is why pyramid of biomass will be smaller. Okay, going upwards. Whereas when you talk about aquatic ecosystem, there the pyramid of biomass is inverted, so you should be knowing it. Okay, inverted biomass. Why? Because phytoplanktons are quite really small. Then you have zooplanktons feeding on them. Then you have small fishes and big fishes. So all the more it gets bigger. So it will be inverted pyramid of biomass in the case of aquatic ecosystem. Very much important. You might have to draw all of them. Now pyramid of energy is always upright. It is going to be like this because it is going to lose 10% every now and then when it is going to go upwards. Cool. So please know that. I think with that we are going to sum up the whole thing. So these are the very important diagrams, my dear children. As I said, please refer the other video also. There is a longer one, but it has all the information until sexual reproduction in flowering plants, whatever important, and I have put the high weightage topics over there. So if you have not watched that particular video, you should be definitely giving it a watch. That is all the more very important. Okay, that will give you more marks probably. And do let me know if you have any more queries or if you want any of my help. I will be so happy to do it, and do let us know if this particular video also was helpful for you. That's all, my dear children. So I hope you're ready. If you're ready for all the diagrams, for practicing all the diagrams, then please put down yes, I am ready to practice everything in the comment section, because I'll be really happy to see. What do you have to comment? You have to comment that I am ready to practice the diagrams. Okay, each day one diagram. Each day, children, one diagram. So your slogan should be one day, one diagram. ready for one day, one diagram challenge. I challenge you from my side. So I'm challenging you for your own good only. If you win this challenge, it will be for your own good, but I will be also really happy. I am ready to lose up over here. Okay, so I am challenging you to practice one day, one diagram that will help you do wonders on the day of your examination as well. So small task, no? Whenever you're getting sleepy and all, you draw this. So that's all, my dear children. I hope this video was helpful for you, and if you like this video, like it, comment down and subscribe to our channel. Bye bye. Take care.